go.
Okay, good morning, and thank you for the invitation. I'll try and do this in 8 to 10 minutes. This is looking at molecules and biomarkers and carbonate concretions from Lagerstätten deposits. There's a whole host of co-authors and students who I will mention throughout, just to acknowledge their contributions. So carbonate concretions, these occur in Western Australia in the Canning Basin, up here, and they weather out of reef basin onwards. Many of you are aware of these famous gorgon nodules. Now these concretions, as we know from the work of Kate Nistick, John Long and Derek Briggs, who look at these in terms of preservation. These ones in the Canning Basin are Australians Lagerstätten, showing the presence of both bone and muscle in 3D preservation, and muscle has been replaced and preserved as apatite, shown here. So these, I'm sure Kate will talk about those this afternoon. Now, before I talk about biomarkers, I just want to introduce you to some of the organisms we look for in geological samples. These occur in the modern day Black Sea. This is the modern day Black Sea, where we have anoxic bottom waters. We have organisms, green and brown sulfur bacteria, which carry out photosynthesis using toxic levels of H2S to fix CO2 in the presence of light, different to normal photosynthesis using water. And in doing so, they make very specific carotenoid pigments and bacterial chlorophylls to capture that long wavelengths of light. So therefore, these organisms indicate hydrogen sulfide in the light zone of the water column. So in geological samples, we look at molecules or biomarkers which derive from pigments, such as this isorenaretine. This is the biomarker of that. Don't worry too much about the structure, just look at the skeleton. Here we have bacterial chlorophyll C, D, and E, which gives rise to polar compounds, which have been reported in geological samples as well, which are indicators of green sulfur bacteria. Whoops. Oh, okay. Now, looking at one of these gorgon nodules, this is some early work done back in 2013 and 14 by Ines Melendez, who took one of these carbonate concretions from the Canning Basin. This was an invertebrate Comutia. It wasn't classified. This has soft tissue preserved in this concretion, and she actually looked at biomarkers to tell us anything about fossil preservation. The fossil here is shown on the right. You can see from the molecular fingerprint it is quite different to the carbonate matrix. You see the carbonate matrix is largely comprised of straight chain compounds, and the fossil has lots of nice steroid components. Now, where do steroids come from? Well, if we look into this, the tissue or the soft tissue of this fossil, we see very high amounts of this molecule called cholesterol, which actually comes from cholesterol, which you find in prawns and crustacea. Based on the percentage of this biomarker cholesterol, we're able to interpret this organism as a crustacean because there's the high percentage of the cholesterol which comes from the cholesterol. So we could use molecular geochemistry as a chemotaxonomy tool. Furthermore, we did the isotopic analysis of this, and we can tell you about the different molecules which come from phytoplankton, the cholesterol coming from the cholesterol, and from the phytoplankton, the diet of the crustacean. Those values are consistent with a source from algae, and the difference between various molecules, we can go down to levels of looking at biosynthesis, but I won't have time to talk about that today. The alkanes which are in the matrix are very, very depleted in carbon-13, and these are interpreted as being from microbial activity of sulfate-reducing bacteria, which are recycling this material and building the concretion. So therefore, Ines looked for the presence of molecules of green sulfur bacteria and found these only in the soft tissue, which indicates that hydrogen sulfide was in the light zone of the water column, when this organism was preserved. So this is how this organism was perhaps killed because of the hydrogen sulfide in the water column. However, when the organism died, sank through the water column, and the hydrogen sulfide actually helps to preserve the soft tissue. The role of microbes is very significant here, and we find the remains of those biomarkers in the soft tissue. Then the organism sinks to the bottom of the water column and is anaerobically degraded by the sulfate-reducing bacteria which produce the H2S in the first place. Alkalinity drops and the calcium carbonate concretion builds up around the carcass. So this is some early work. So we've also looked at, given that these samples are very immature and potentially may contain intact cholesterol, there's very few reports of sterols in geological time, but we've gone back to the Paleozoic here and in the Devonian, and indeed we found sterols. We found 70 different steroids in the fossil alone, which represent all the cholesterol and intermediates that occur actually in the water column, through to compounds which we generally find in petroleum, which are associated with maturity, but in this case are probably all microbially mediated from cholesterol through to sedimentary components. So we found the diagenetic continuum of sterols through to the components we find in oil. Now moving to another example, we have the work of Chloe Plett, looking at a vertebrate for an ichthyosaur from the Jurassic, 183 million years old, from the Posidonia shale. We have a pure calcite body, we've got a pyrite rim and traces of quartz, and we have the ichthyosaur bone here. The vertebrate bone contains very nice corticular bone, very hard bone, and spongy trabecular bone, which is consists of sparicalcite and barite, which has filled in the pores of this bone. Looking further through SEM imaging of the trabecular bone after light acetic acid treatment, you can see the presence of collagen fibrils in the form of fluoroapatite and these tentative red blood cell structures as well present in the bone. These red blood cell structures are about 4 times smaller than those in modern organisms, and I'll come back to that. We also can do TEM and SEM imaging of these materials and confirm that this has organic material in it, and this is some of the sterols which we found in the bone. In the bone there's high amounts of cholesterol of the diet of the ichthyosaur, along with other sterols which are from the algae, and there's an isotopic difference between those, which is consistent with a higher order of the food chain for the cholesterol, which is in the ichthyosaur, which it has formed from the dietary sterol ethyl cholesterol in algae. Furthermore, we find the whole suite of steroids that we found in the Devonian crustacean as well. We find the whole suite of steroids in these samples, so that's consistent. Also in the bone we find the presence of breakdown products of hemoglobin, malamides, and their isotopic composition is consistent with a heme source. The ichthyosaur lived during a time when there's very low oxygen levels in the ocean and atmosphere, and probably made these very small red blood cells to be able to effectively transport the oxygen around the body. So this is the ichthyosaur and the red blood cells. And current work we're doing is actually working on different types of concretions. These come from Mazoncrete, which is a siderite concretion, iron carbonate containing exceptionally well preserved plant tissues. We find some interesting components in these samples which relate to the cutine composition in modern fruits. This is a cutine composition we find in an extinct gymnosperm or extinct seed fern, actually has the same cutine composition as those found in tomato and strawberries, the seed fruits. So we think there may be an evolutionary link between our seed fern and modern angiosperms. So this is where we're going. This is just a start of a big project on concretions and what information they tell us using molecular geochemistry. And we're working on the end Cretaceous event as well, which I don't have time to talk about today. But yeah, this is us at Curtin University. And thank you for listening to an organic geochemist about molecules.
Thank you.